day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled UVC Treatment, a new procedure for viral inactivation. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for up to 90 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using this chat panel. And at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically for you. Please note that this event will be recorded and made available for future download. And at this point, I would like to thank Eurofins Lancaster Laboratories, who has helped develop the content for this presentation. With a proven track record of providing quality scientific solutions for more than 1,000 pharmaceutical and biopharmaceutical companies in the world, Eurofins Lancaster Laboratories is a global leader in biopharmaceutical laboratory services, providing innovative and timely scientific solutions to streamline the drug development process. With facilities in Lancaster, Pennsylvania, Portage, Michigan, Dungarvan County, Waterford, Ireland, and Munich, Germany, and a global capacity of over 400,000 square feet, Eurofins Lancaster Laboratories has the capabilities to meet global regulatory requirements. All of their facilities offer CGMP-compliant laboratory services and operate under the same strict quality control program and utilize the same LIMS system. And now I would like to introduce our first speaker for today's event, and that is Kate Bergman, PhD. Dr. Bergman is a manager of viral safety and viral clearance services at Eurofins Lancaster Laboratories. She has extensive experience in cell line characterization, lot release testing, and viral TSE clearance validation, and has numerous publications and presentations in the field. She has performed biomedical research in protein chemistry, cell biology, and virology at Georgetown University, Scripps Research Institute, and Rockefeller University. She received a PhD from Brown University and a Bachelor of Arts degree from Mount Holyoke College. Dr. Bergman served on the PDA task force on preparation of virus spikes used for virus clearance studies. And our second speaker for today is Todd Tellerico, PhD. Dr. Tellerico is Vice President of Manufacturing for Medicago and oversees the U.S. and Canadian development and manufacturing operations. He has been responsible for Medicago's USA's completion of an aggressive design-build project resulting in rapid construction and deployment of a facility designed to produce more than 10 million doses in 30 days of H1N1 vaccine, expressed in and purified from tobacco plants. He began working in the vaccine field with AlphaVax, where he was a senior director responsible for development and process scale-up of a virus replicon particle vaccine. And our third speaker for today is Sherry Dolan, Field Marketing Manager, Virus Clearance Technologies at Sartorius Steady in North America. Sherry Dolan has been at Sartorius Steady North America in the Purification Technologies group since 2005. Her responsibilities as Field Marketing Manager include supporting small virus removal membranes, membrane absorbers, and UVC technology. Throughout the past 25-plus years, she has held a wide variety of positions in the biopharmaceutical industry in both large pharmaceutical and small biotech companies. With a focus on downstream purification, her previous positions range from R&D, process development, clinical manufacturing, field application support in the areas of cell culture clarification, chromatography, filtration, and final filling operations. And now, without further ado, I would like to hand the mic over to Dr. Bergman. Dr. Bergman, you can begin your presentation when ready.
Thank you, Andrew. I'm going to start by giving you some information on the inactivation of virus by UVC treatment. Biopharmaceuticals sourced from either animals or human systems are at risk of contamination from viruses. And the contamination could come from source material, cells, tissue, blood, things like that, from the raw materials used in manufacturing, and from the manufacturing process itself. Viral contamination events are known to have occurred in the past. They have occurred with envelope viruses, which are viruses that have a lipid envelope. The most common example has been Cache Valley virus infections in CHO cells, and there are at least 4 known events of this type. However, most industry viral contamination events have been due to non-envelope viruses. That's viruses without a lipid envelope. There's at least 15 known events involving viruses such as reovirus, MMV, vesivirus, PCV, and EHDV. The strategies for assuring the absence of virus in the final product include control and testing of raw materials and master and working cell banks, as well as testing of the unprocessed bulk product for viruses. And third is to include in the purification process steps that have the capability of removing and/or inactivating viruses. And these steps in the purification process are evaluated by means of viral clearance studies where model viruses are used to evaluate the effectiveness of the process. Virus removal steps are steps where the virus is removed from the process but is not necessarily inactivated. Example steps include virus filtration, column chromatography, and precipitation steps. Chromatography and precipitation are not considered robust due to the variation in performance with different viruses and variation based on changes in condition. Some steps also may show viral inactivation as well as removal. Viral inactivation steps are steps where the virus is inactivated or killed but is not necessarily removed from the process. Examples are low pH heat, solvent detergent, and irradiation. The performance of these steps does vary with different virus types. The steps are usually considered robust within certain ranges. And regulatory agencies usually expect to see at least one effective inactivation step in a purification process. Now, viral inactivation steps are generally more effective for the envelope viruses than they are for non-envelope viruses. For example, for low pH treatment, a lower pH is required for inactivation of non-envelope viruses. For heat treatment, higher temperatures and longer times are required. Solvent detergent treatment is just not effective for non-envelope viruses, and irradiation requires higher doses to inactivate non-envelope viruses. And the product often cannot tolerate these extreme conditions that are required to inactivate the non-envelope viruses. So there is a need for an effective inactivation procedure for non-envelope viruses. UV irradiation has been known since the 1940s to be effective at inactivation of viruses. However, since it causes protein damage, it's not been traditionally used in the biopharmaceutical industry. And early UV technology was unsuitable because the penetration of the UV radiation is low and the reactor design did not provide a uniform dose throughout the volume of material. And also because the reactor design did not allow for representative downscale models. However, more recently, improvements have been made in UV technology. In first place, monochromatic UVC light has been used, and that is at a wavelength of 254 nanometers, which targets primarily nucleic acids rather than proteins. And this results in minimal protein damage. Reactor designs have been improved to allow efficient mixing of the product stream, and this provides a uniform UVC irradiation. And with this improved technology, it's been found very effective at inactivation of parvoviruses, and that greater than 4 logs clearance can be observed. The technology we'll be talking about today is the Uvivitec technology from Bayer Technology Services, marketed by Sartorius Stedim. It uses monochromatic UV light at 254 nanometers. The liquid is pumped through a reactor, and the helical reactor tube provides efficient mixing and a very short path length. And in this instrument, the dosage is controlled by the light intensity and the flow rate through the system. Now this slide shows the wavelength of the absorption of irradiation at different wavelengths, and shows that the 254 nanometer wavelength is very near to a maximum of the absorption by nucleic acids and to a minimum absorption by proteins, although you should be aware that the absorption by proteins is not zero. In the panel on the right, you can see that the radiation is very nearly monochromatic. So at Eurofins Lancaster Labs, we've performed some studies to evaluate the effectiveness of viral inactivation. We've used a model protein solution, bovine serum albumin. We've used viruses from a number of different families. We've looked at a range of UV doses, and we've looked at varied treatment conditions, including protein concentration, different buffers, and different pH conditions. This is the panel of viruses that we've been working with. They range from small viruses as parvoviruses, MNV and PPV, up to large virus, pseudorabies. We have RNA and DNA viruses, single-stranded and double-stranded, enveloped and non-enveloped, and with a wide range of genome sizes as well. So this is our initial studies looking at effective different doses on all of these viruses. We were using a constant buffer condition and a constant protein concentration here. And you can see that the two curves at the top represent the two parvoviruses, and we're seeing above 6 logs of clearance, even with very reasonable UV doses. In the middle, there's another group of viruses that are intermediate reactivity, and then there's 3 more viruses down at the lower end that are not very sensitive to UV irradiation. So the most sensitive viruses are MMV and PPV. Intermediate viruses, bovine viral diarrhea virus, polio, and pseudorabies. And this group itself spans from very small to very large, so that shows you the variability. The least sensitive viruses are murine leukemia virus, adenovirus, and reovirus. Now, looking at the sensitivity against the characteristics of the viruses, there is no correlation with the size of the virus particle. There is no correlation with the genome type or the size of the genome or the strandedness of the genome. And there's no correlation with whether or not the virus is enveloped. Given the fact that the mechanism of action of UVC is creation of pyrimidine dimers, it is possible that the sensitivity to UV may be based on frequency of sequential pyrimidines. Now, the 3 viruses that are the least sensitive here do have additional mechanisms by which they can escape the effects of UV irradiation. Xenotropic murine leukemia virus, as all retroviruses, contains 2 copies of the genome within the virion. So if one genome is inactivated, the other genome may be able to complement the failure and thus escape inactivation. And for both reovirus and adenovirus, there's a phenomenon called multiplicity reactivation. And this is a phenomenon where if multiple virions infect the same cell, they may complement within the cell and again provide an escape from the UV inactivation. And these reasons may explain why these 3 are the least sensitive of the group. Now, looking at different buffers and pHs, we're looking here at 3 different buffers. One is acetate pH 5.5, PBS pH 7.2, and then tris buffer pH 8.5. And here with MMV, you can see that there's no significant difference in the dose response in these 3 different buffers, again with a constant protein concentration. For reovirus, again, no significant difference in response. And for xenotropic murine leukemia virus, again, no significant difference in response in the 3 different buffers. Looking at effective protein concentration now, we selected, for each of the viruses tested, we selected a dose that showed partial inactivation but not complete inactivation, and we repeated the study in different protein concentrations. And you can see that, again, there's no significant impact of change in protein concentration over the range from 5 to 30 milligrams per mil. So in summary, There's no effect of buffer or pH on the sensitivity of viruses to UVC inactivation. There's little or no effect of protein concentration on the sensitivity to inactivation. And therefore, UVC irradiation is a highly robust process that can be performed under a wide variety of process conditions, making it a very versatile step to achieve virus inactivation. Now I'd like to acknowledge my team here at Lancaster. Stephen Stoltzwitz has been the primary mover behind organizing and designing the study, and Katie, Caitlin, Brian, and Nick have all contributed to the assays and performance of the study. And I'd like to also thank Sherry Dolan at Sartorius Stidine Biotech for her support in helping us achieve this. So this completes my part of this presentation, and I'm going to turn the mic back over to Andrew, who has a question for everyone.
Great. Well, thank you, Dr. Bergman, for that. We certainly do have a question for the audience right now, and you can vote on this question in real time. The question we have, are you currently using UVC technology for, and you can select any of the answers here on the screen by clicking, is that upstream treatment of raw materials, downstream during purification, or are you not using UVC yet? That question again, are you currently using UVC technology for one of the following answers on your screen there? So it looks like most of you have clicked on your screen and voted, so I'm going to close polling now and share the results with you. It looks like the vast majority of you, 89%, say that you're not using UVC technology yet. That's followed by 11% saying downstream during purification. And with that, I'm going to hand the mic over to Dr. Todd Tallarico. Dr. Tallarico, you can begin your presentation when ready.
Great. Thank you very much, and good afternoon. I've been asked to present to you a case study of using UVC in a process to produce a plant-derived vaccine. And I'd like to begin by just reviewing a couple details about the vaccine we're making in Medicago. We are indeed producing vaccines in plants, and it's a relative of tobacco. And the vaccine we produce for a flu indication is actually a virus-like particle. So the vaccine itself is large and similar in size to a virus. You can see that the VLP ranges in size from about 120 to 170 nanometers, depending on which strain of HA protein is expressed and contained in the VLP. The VLP itself is a lipid bilayer that has in it inserted the HA protein, and the cartoon there shows the Medicago VLP with green spikes on it, and it contains only the HA protein, as opposed to the flu virus itself, which has the HA protein. It has a neuraminidase in there. It has some matrix proteins, and also contains the RNA, which provides for the infectivity of the virus itself. The VLP does not contain any genetic material in it. So again, it is a nice target for UVC. And at the bottom, you'll see an electron micrograph of the actual flu virus. It's an H1N1 California strain, and next to it is a picture of the VLP that's expressed with that same HA protein containing H1. And they're very similar in size and structure, and you can see the spiked buds on the outside of it. So the particle itself, again, the thing to remember here is it's large and very much like the influenza virus. Medicago has produced a number of different VLP with a number of different HA proteins in it. Some of these have been in clinical trials. In Canada, we've done some pandemic work with H5. We've done a phase 1 trial in the US with an H1, and we're currently have a trial ongoing with a quadrivalent that contains 4 strains of that, and we've recently been approved to start an H7 trial in Canada. So we have experience with this, and so we're able to look at a lot of different kinds of VLP in models. So if we think about the vaccine and some of the challenges that we have in thinking about viral inactivation, and let me just add here, we really thought when we started this, being a plant-derived vaccine, it's inherently safe. There are no human viruses that can replicate in plants, and no plant viruses that can be transmitted to humans that have ever been reported. So, we thought maybe we would be exempt from having to have viral clearance in the process. But, as we talk to regulatory agencies, they've expressed the wish that in fact we do have some kind of either an activation or clearance step in the process. So we've embarked in challenging the process and asking, can we get viral clearance in there? Right off the bat, as many of you would suspect, using a filtration step to remove virus from a VLP process doesn't get you anywhere. You take away all your product as well. So removal is out by filtration. We looked at the chromatography steps that we currently use in production, and we found that they were ineffective in removing spikes of model viruses. Typically less than 2 logs removal we're seeing, and as you heard from Kate, these aren't necessarily robust steps, but it's nice to be able to rely on them. We found that we weren't getting a lot of removal there. We looked at formaldehyde addition to the process, and what we found was that if we were going to not impact the product, the kind of contact time and everything we used didn't also not result in much removal or inactivation of virus in the process. And then when we actually spoke to the FDA, they said, we'd be happier if you weren't using formaldehyde in the process at all. So that was a good guidance to try to get formaldehyde out. We took a look at gamma irradiation as a terminal sterilization step and found out that it pretty much killed the activity of the product from a potency standpoint, as well as some of the other key quality attributes of the product. So we thought, well, let's take a look at UVC. And when you think about using UVC in your process and with your product, you have to consider any impact it may have to the protein. In our case, we have to also think about impact to the lipid and into the particle structure itself. Of course, as a vaccine, we worry about anything that might happen to compromise immunogenicity. And finally, from a quality standpoint, you don't want to be doing anything that's going to mess up the stability of your bulk drug substance or your product. So those are the things we considered, and we looked in the literature. And when you look out there, you find that, in fact, at certain doses, you see impact to protein. At certain amino acid residues, you see some disulfide damage in thiols. And here's a couple examples of what you can find if you look in the literature. There is a pretty long history of people of at least looking at these kind of things. So we took a look at what kind of impact it might have to our protein. And initially we looked at H1 and H5 VLP as our models. And our initial thought, based on some preliminary studies, was we would shoot for a dose of around 500 joules per square meter. So in these studies, we also added a couple really high doses so that we could be certain if there were impact to the protein, we'd see it. And so you'll see there we have 500, 1,000, and 2,000 joules per square meter of the UVC doses that were studied. And what we found was that especially at the high end, we could see a dose-dependent decrease in the SRID value, which is an indicator of the potency of our product, and also the ability of the product to hemagglutinate red blood cells. We also observed slight increases in particle size and polydispersity of the mix as we increased the UVC dose. We didn't see a change in total protein concentration, and we have yet to determine what impact this has on long-term stability. Those studies are underway right now. So in addition to the H1 and H5, we looked at some other VLP at lower doses, and I'll explain that more in a second. So here's a figure that shows for the H5 HA protein the impact, and the x-axis contains increasing dose of UVC, and on the y-axis you'll see basically a concentration either of protein or an SRID value, which is the potency indicating one, and on the other axis you see basically the hemagglutination dilution. It's a twofold dilution type assay. And you'll note that the triangles and the red squares indicate that yes, as we increase the dose, we really do see something happening to this particle. However, the blue diamonds, the total protein, remains unchanged across the dose regime. And this, again, is repeated for H1, and we saw the same trends here. A drop in HA dilution value and the SRID, while the protein was held, remained fairly constant. We then took a look at a couple lower doses, and here you'll see a 250 joule per square meter dose and a 350 joule per square meter dose on a different VLP. This was a B Brisbane VLP we had. And and we compared the numbers you'll see in this table, or it represents the percent change compared to the untreated control. So you'll see total protein, SRID, HA dilution, DNA and turbidity really didn't show much change at these dose levels. The size of the particle, when we looked at dynamic light scattering, again, not much change. In the polydispersity, or like the distribution of the different sizes in there, again, not changed too much in the control. A different way to look at size is nano-tracking analysis, and we thought, maybe at the higher doses we were seeing a little bit of change, increase in size, but again, it's hard to say that's statistically significant for this technique. And finally, no change in pH. So, yeah, we had a pretty good idea that, while our initial thought was let's shoot for 500, maybe we need to back off of that a little bit and look in the 250 to 350 joule per square meter dose regime. And we also caught on a couple additional techniques, and these are really developing assays. But our analytical and process development folks are great. They know a lot about these particles, so they said, hey, let's take a look and see if we can see a change in the surface charge on the particles. And the way they did that was they used cation exchange chromatography, and they said, as we expose things, do we see a change in retention time or peak shape with this molecule? And you'll see on here, the blue line represents something that wasn't exposed to UVC, and then the other colored lines show increasing doses. And when you take a look at it, you can see the elution conductivity or retention time does increase over time as you give higher and higher doses of UVC. Now, they're not huge changes, but it indicates that something is going on at the surface of the particle. And you can also see that the peak shape changes. Could this be aggregation or something? We don't know, but certainly it is a change, and we have yet to go in and delve in through peptide mapping or anything to find out exactly what residues are being changed. But we can see there is some impact there. Finally, because our particle contains a lipid bilayer, we wanted to ask the question, are we seeing lipid oxidation? And again, with an assay that was an off-the-shelf assay to look at lipid oxidation, we could in fact see slight oxidation of the lipid going on as the dose was increased. So again, we don't know if this really impacts anything, but we know it's there, so it means we'll look further into this issue. A big key for us is if you expose the particle, do you change the immunogenicity? And so there have been a lot of studies in rodents with these particles, and two are shown here. And let me point out, these are performed on process intermediate. So the process intermediate is taken, exposed, and then diluted and injected into the animals directly. And you see for H5 VLP that it appears as you start to increase the dose of UVC that these are exposed to, that you mute the immune response. And H5 VLP is really a tough one. It's not very immunogenic, so you always give this with an adjuvant. And so we said, let's take a look at another VLP, H1, which tends to be a little more immunogenic in animals. And you can see in the case presented below that, while the 500 joule per square meter dose doesn't seem to have a big impact on the particle, as you start to move out towards 2,000 joules per square meter, the impact is clearly seen, that the immunogenicity is muted somewhat. We took a look at H5 VLP then again with lower doses, and 200, 350, and again right at that 500 cusp. You can see that nothing statistically significant stands out, that there's a difference in there. So, maybe in the 350 joule per square meter dose range, that we have a nice process step for viral inactivation. And while these are process intermediate, if you look at the drug product itself after it's gone through further purification in the process, and we look at the H5 VLP here, you can see some material that was used in a phase 2 study on the right, its response as compared to similar product that was exposed to UVC dose of 350 joules per square meter. Again, no difference in what we see in immune response in animals in this case. So, further suggestion that the 350 joule per square meter dose may be all right, especially on drug product. All right, so while that's the impact of the treatment to product, we also are interested in, would this result in good viral clearance for the process. So as you heard from Kate, we used model viruses, we spiked into process intermediate, and this was process intermediate was produced either in a pilot operation or full scale as per batch record. So just the way we would produce it if we were making it for human use. And we compared the intermediate that was generated there to previous lots to confirm that the quality was representative of what a typical lot would be. And we then moved the material to contract laboratory and did the spiking studies. And here I'll give you a few cases. One of the first ones we did was product intermediate from B Brisbane spiked with either the MMV or the XMULV. And as parallels case results, the XMULV is not a great virus for UVC inactivation. We got almost 3 logs, but it took a dose of 750 joules per square meter. However, the MVM was completely inactivated to the sensitivity of the assay, so we could detect no virus in the output from there. So again, greater than about 5 logs of inactivation there. We then moved on to an H7 process intermediate and asked about a few different viruses. So we looked at Rio, pseudorabies, and BVDV. And again, in this case, every time we looked at the dose of 200, 350, or 500, the BVDV and the pseudorabies was inactivated to the extent that the assay could detect. So we saw complete activation there, about a little over 4 logs for the pseudorabies and more than 5 for BVDV. The Rio, at low doses around 200, we didn't get 0.4. We got almost 6 at a dose of 350, and at 500 we saw complete inactivation, so more than 6 logs. So again, we were very happy with the results here, and it spurred us to go more. And so we took a look at H5 VLP also in support of our pandemic process in Canada, and we have MMV again here, pseudorabies, and BVDV as a backup. We saw complete inactivation of BVDV and pseudorabies at all doses, 200, 280, and 350. And then in this case, we did not get complete inactivation of pseudorabies, but we saw almost 4 logs inactivation at a dose of 350. So our conclusion from all this was if we use UVC and take high doses in with our product, we definitely can see an impact to the HA protein, decrease in potency, increase in particle size. Is this aggregation? Not sure, but we certainly see it increase. We know there's a change in the surface charge and probably some impact to lipids as well. Doses of approximately 350 joules per square meter appear to be appropriate for the VLP, but we still need to complete the stability studies long-term to confirm that this is true. The UVC has proven to be very effective at inactivating a broad range of viruses in our matrix, and the retroviruses were the ones that were the least inactivated. And UVC looks like it's a nice alternative for viral inactivation, especially for a VLP vaccine, because one, there's no nucleic acid in this that is needed for immunogenicity or immune response, and 2, it's such a large particle, and it's so closely related to other viruses that typical removal steps in the process aren't great candidates. So the UVC looks like it, in fact, is quite nice. And with that, I will hand the presentation to Sherry Dolan, and she will tell you more about the system.
Thank you, Todd. The data that has been presented so far has been generated using the Sartorius UViviTech lab-scale UVC inactivation system. So this presentation will give you an overview of the system properties, and I'm going to try to describe the unique features of the system, which makes it suitable to treat a wide range of biopharmaceutical products. In October of 2008, it was announced that Sartorius Stedim Biotech and Bayer Technology Services, which are both located in Germany, had signed an exclusive cooperation agreement on the manufacturing and worldwide marketing of UViviTech products. The innovative design of the system allows for effective inactivation of viruses, especially the difficult to inactivate parvovirus, while maintaining product integrity in a variety of biopharmaceutical products. Therefore, the UVC technology may be especially suitable as an alternative or supplementary method for both upstream and downstream virus inactivation. So Sartorius offers several products to support orthogonal virus clearance technologies. The UViviTech product was the first inactivation method to be added to this platform. Another inactivation method we support is low pH, which is inactivating envelope viruses. We have an automated system to perform this, and the system is called the FlexAct VI. The second orthogonal technology is virus removal by adsorption, which utilizes membrane chromatography. So viruses will bind to the membrane, and the product will either flow through the membrane or the product can be eluted from the membrane. And the third orthogonal technology is filtration membranes, which physically remove viruses greater than 20 nanometers. Currently, Sartorius offers 3 different virus retentive membranes, along with a new pre-filter, which was specifically designed to increase the throughput and decrease the flux decay on these membranes. So the UViviTech system is based on inactivating viruses using UVC light. UVC light has a wavelength of 200 to 280 nanometers and is not dangerous to humans' health. Overexposure causes skin redness and eye irritation, but not skin cancer or cataracts. Light in this wavelength range is known to be an effective inactivation method for air, surfaces, and water, and as Kate has mentioned, the technology has been around since the 1940s. The mechanism for virus damage is the penetration of UVC light into the nucleus, and this causes the formation of thymine dimers in the nucleic acid. So all types of viruses, whether they're DNA, RNA, single-strand, or double-strand, may be susceptible to this damage. However, as Kate has shown, viruses will have different levels of sensitivity, and parvoviruses seem to be the most sensitive. UVC light is also known to cause protein damage. Proteins are sensitive to UV light and may be damaged mainly by two mechanisms. The first mechanism is oxygenation, and the second is disulfide photolysis. Therefore, when utilizing this technology, it's important to test your protein for damage when it's subjected to different doses of UVC light. And Todd has shown a very good example of how to go about testing your product for UV sensitivity. In addition to what Todd has shown for monoclonal antibodies, the following tests have been shown to be useful to determine protein integrity after UVC treatment. You can use high-performance size exclusion chromatography, reverse phase chromatography, ion exchange, capillary electrophoresis, peptide mapping, free thiol quantitation, circular dichroism, and bioassays. So as Kate had mentioned already, the UViviTech system is based on using a low-pressure mercury lamp with a fixed wavelength of 254 nanometers. And this specific wavelength was chosen due to the high absorbance of RNA and DNA, which causes the damage that I just mentioned, and the relatively low absorbance of proteins. So the main goal and challenge of this technology is to damage your viruses while maintaining the integrity of your protein. The traditional annular UV irradiation systems, such as those used for water treatment, which is shown on the left side of the slide, may not be suitable for protein solutions. UV radiation decreases exponentially with the distance from the light source, so the fluid which is closest to the light receives a high intensity, while the fluid farther from the light receives a low intensity and possibly none at all. In these systems, prolonged exposure times and high doses are required and may result in protein damage. So for instance, in order to sanitize drinking water, a dose of up to 10,000 joules per square meter may be used. So in contrast, as you've heard, the typical dose needed to completely inactivate parvovirus using the UViviTech system is around 100 joules per square meter, or 100 times lower. The innovative Bayer Services reactor, which is shown on the right side of the slide, has been designed specifically to eliminate these issues with the annular reactors and by utilizing a novel helical flow channel. So here you can see the difference in residence time distribution for an annular reactor, which is shown by the blue line, in comparison to the narrow residence time for the helical reactor, which is shown by the red line. As the curves illustrate, the annular reactor will have areas of low residence time, which results in low virus inactivation, or may also have areas of high residence time, which may cause protein damage. So the helical reactor has very efficient mixing. This allows for a narrow residence time and also ensures complete virus exposure and limits protein damage. This will be further described in the next slide. So in the Bayer Technology Services reactor, the basic idea of the system consists of a helical channel tube format with a semicircular outer side consisting of Teflon. And you can see that on the left side of the screen. This has an inner surface of quartz glass. Here we see the inner surface. So the product will actually come in contact with the Teflon and the quartz glass. Inside of this module, you will see a rod-shaped UV light source, and this light source slides inside of the quartz glass. The novel helical channel induces Dean vortices, and this provides for highly efficient mixing in the fluid stream and optimizes virus exposure to the light source. As a consequence, the dose of UVC irradiation can be delivered evenly and uniformly throughout the solution. Therefore, the required residence times in the reactors are extremely short. For instance, for the lab scale system, it's about 20 or 30 seconds, and the UV exposure can be accurately controlled. Since the formation of Dean vortices is critical to properly control the dose, it's very important to operate the reactors within the specified flow rate range. Operating the modules at a lower or a higher flow rate than recommended will reduce the Dean vortices, and that will impact the performance of the system. So for scale-up, the UViviTech system is available in two sizes. On the left side, you will see the lab scale system, and this was a system that Kate and Todd had used for their studies. This is a bench scale system, and it may be used for scale-down validation studies, process development, as well as treating small batch sizes of product. The irradiation intensity is 60 watts per square meter, and the flow rate range is from 6 to 20 liters per hour. The system shown on the right is a custom-built GMP process scale system. These systems have an irradiation intensity of 194 watts per square meter and a flow rate range from 30 to 120 liters per hour. For these systems, you may use up to 4 modules in parallel, which allows for a maximum flow rate of 480 liters per hour. And these systems may be used for product batch sizes up to 2,000 to 3,000 liters. So UVC dose determination. The dose is determined by the multiplication of the mean intensity with the mean residence time. The mean intensity is a function of the absorbance of the product at 254 nanometers and the lamp power. The lamp power may be slightly adjusted on the lab scale system, however, it is fixed for the process system. So the absorption of the product at 254 nanometers is a very important parameter, and to achieve a relatively low dose, it's optimum to have an absorption value between 5 and 22 absorbance units. This may be a factor in choosing where to place the UV treatment within a process, and Kate will talk about this a little more when I'm finished. The residence time is directly related to the flow rate, and as mentioned, it is important to be within a specified flow rate range where the formation of Dean vortices is optimal. Now, in order to calculate your dosage, we have an Excel spreadsheet where you can enter the absorbance of your product and the dose you want, and the spreadsheet will tell you what flow rate you need to use. So you don't need to plug any values into this equation here. We have a simple spreadsheet that helps you do that. So the mode of operation, if a protein solution has less than 22 absorbance units at 254 nanometer, a single pass operation may be utilized, and this is the most common mode of operation for the system. However, if you have a highly absorbent solution, which is greater than 22 absorbance units, it may be necessary to perform either multiple passes through the module or perform a recirculation, as shown on the slide. So robustness of scale-up. The scalability from the lab scale system to the process scale system has been proven using two methods. The first method consists of performing a dose versus inactivation curve for bacteriophage MS2, which is a model virus for parvovirus. And as you can see on the graph, the inactivation kinetics are similar for both systems. For example, we can see that at a dose of 100 joules per square meter, there's very similar inactivation for both the lab scale system and the process scale system. As the dose is increased, the kinetics remain the same. And a similar robustness of scale-up study has also been performed using fluorescent microspheres. So in summary, the novel helical reactor allows for uniform radiation by utilizing radial mixing based on Dean vortices. The applied dose range is restricted to the absorbance of the product and the flow rate of the system. Since proteins are sensitive to damage caused by UV light, it's important to conduct protein stability studies, and the UVC systems are robust and scalable. So this concludes my portion of the webinar. Thank you, and I will pass this along to Kate.
Thanks, Sherry. So now let's consider for a little bit the use of UVC in a purification process. As we've shown, UVC is a critical addition to purification process for products such as VLP, where it's very difficult to show sufficient viral clearance. And Todd's data demonstrates this very well. We've also found it can be useful for products like IgM, where other process steps that are commonly used, such as viral filtration and low pH treatment, are not available due to the size or the fragility of the molecule. However, UVC can also be useful for other products, even including monoclonal antibodies. It can provide an additional clearance step, especially for parvoviruses where the clearance is limiting. And it can also be used to replace a more complex step and actually simplify the process. There are very few critical process parameters for UVC treatment, and it's very simple to perform. So it's easy to integrate into the process. Now, and looking at this slide again, you're looking at the only critical process parameters there are aside from the product itself, and that's the absorbance at 254 nanometers, the power of the lamp, and the flow rate of the system. So as you can see, there's not a lot of things that have to be evaluated in order to integrate this into a process. Now the dosage is dependent on the absorbance of the material, as Sherry has said, and lower absorbance results in a higher dosage. So the absorbance is due primarily to nucleic acids, but proteins can also contribute as they absorb at a lower level. So in the absence of nucleic acids, the protein concentration of less than about 10 milligrams per mil limits the minimum dose obtainable and may require a higher dose than desired. And conversely, in the presence of a high absorbance material with an A254 greater than about 10 or so, it limited in the maximum dose obtainable, and Sherry's data also shows that as well. This is a graph of the minimum dose obtainable at different protein concentrations in our studies. And you can see in the absence of protein, the minimum dose was approximately 400 joules per square meter, and it descends to a minimum at approximately 10 milligrams per mil protein. And after that, the minimum dose is just determined by the maximum flow rate of the system. Now, as we've discussed, all three of us have discussed, there is an impact of UVC on the product. The proteins do absorb UVC at a low level and therefore can be impacted by it. The damage is dose dependent, and a common mechanism in the macro scale is aggregate formation of proteins. However, work has been done to date shows that doses of 200 joules per square meter or less are generally tolerated with low levels of aggregate formation. In one example of an IgM product, the aggregate level increased to approximately 4%, but the polishing chromatography steps were able to remove the additional aggregate, so this was considered satisfactory. So integration of UVC into purification process is relatively easy because it's a flow-through operation. It can be considered kind of like a plug-and-play where you just connect two tanks by the system, and that's all it takes. It's not impacted by buffer conditions or protein concentration, so it's not challenging to find a suitable location to place it. The optimum placement is probably an area of moderate process volume, which controls the amount of time that it'll take to perform the step. Higher protein concentration, so the dose can be easily controlled. It's probably best after the capture step so that the bulk of the nucleic acids are removed. And it's probably best after an upstream polishing step for removal of aggregates. And at this point, I'm going to take a break and let Andrew ask you all another question.
Thank you, Dr. Bergman, for that. We certainly do have another poll question here for the audience that you can vote on in real time by clicking on your screen. That question, would this UVC technology be appropriate for your manufacturing process? And your options, of course, are yes or no. So you can click on your screen right now to vote on that. Again, that question, would this UVC technology be appropriate for your manufacturing process? Looks like most of you have voted at this point. I'm going to close polling and share the results with you. Looks like the vast majority of you say yes at 82%, 18% of you said no. With that, I'll hand the mic back to you, Dr. Bergman.
Thanks, Andrew. All right, so this is a pictorial description of what I just said a minute ago. It's probably not a good idea to place your UVC upstream of the capture step. There'll be a high process volume resulting in a long time to perform this step, and the high absorbance from nucleic acids may limit the UVC dose. And down at the bottom end of the purification process, after the polishing step, so maybe a low protein concentration resulting in higher than desirable UV dose, and there may not be an opportunity to remove any aggregates that are formed. Instead, in the middle of this diagram, in between the capture step and the polishing chromatography steps, is likely the best place for placing the step. The process volume is intermediate, and their polishing steps are able to remove any aggregates that may have been formed. Another use for UVC treatment is to treat raw materials used for manufacturing. This can be used as a barrier treatment for the raw materials before they enter the manufacturing stream. Most of the materials are likely not subject to product damage because they don't absorb in the UV range, so that higher doses can be used to achieve maximum inactivation. And the flow-through mode of the system is compatible with media addition. In conclusion, then, UVC is a highly effective in inactivation of parvoviruses. This is important since it's difficult to inactivate these viruses by any other means. There is a continuum of virus sensitivity to UVC which does not correlate with any obvious properties of the virus. Viral inactivation does not vary with protein concentration, buffer, or different pHs. It does cause product damage, but the amount can be controlled, and damaged product can be removed during later purification. And I should say that it is important to evaluate the kind of damage that you're seeing with UV inactivation before determining your final conditions for running the step. UVC treatment can be placed in many places in a purification process, but the optimum position may be between the capture step and the polishing steps. And this treatment can also be useful in treatment of raw materials. So this concludes our webinar presentation. All of us thank you for your attention, and Andrew will now take over and open the floor to questions. Thank you.
Great, and thank you, Dr. Bergman. Thank you to all of you for those great presentations. At this point, I would like to invite our audience to continue sending their questions and comments in right now using that questions chat window for the Q&A portion of the webinar. I've already received some questions, so I'm going to start with the ones we have here. Our first question, can you speak to the effects on the protein activity if the product contains an enzyme?
This is Kate. Really, the effects can't be known without investigation. It doesn't matter particularly whether it's an enzyme or an antibody or a VLP. You're going to need to look at the irradiation and look at the effects on the activity and performance of the enzyme in order to determine what's an acceptable range. The lab scale units are available from Sartorius to Dean for rent, so you can lease one for a short period of time and do some preliminary evaluation to generate materials that you can analyze and see whether this is going to be suitable for your material.
Okay, thank you for that. Our next question here, what is the relevance of Rio 385 and XMULV? If you could elaborate on that a bit.
Well, all of the viruses that we used in our study are viruses that are commonly used for viral clearance studies. They are model viruses that represent viruses that may potentially contaminate a purification process. XMULV is a model for retroviruses, which are the commonest contaminant that are or commonest potential contaminant in purification processes. Reovirus and adenovirus represent viruses that are usually models of an unknown contaminant, although adenovirus is certainly a very relevant model virus for things such as vectors, viral vectors, and also products made in PRC6 cells, since those cells contain adenoviral sequences. The fact that they're less sensitive to irradiation than the other viruses tested is just a function of the properties of the individual viruses.
Right. Okay. Thank you for that. Our next question here. Is there any value of UVC where penetration is needed, such as with tissues?
Well, as has been seen, the penetration of UV in the UVC range is very low. It can be as little as a millimeter or so. So it's not going to be very effective in materials that are either not transparent or fairly thick, unless all you're looking for is a kind of surface disinfection.
Okay, thank you for that. Our next question, how much effort has been placed into optimization of the geometry of the helical chamber? Do you believe that you have achieved the optimal geometry, or is there room for further improvement?
Hi, this is Sherry Dolan. Bayer Technology Services has been working on the design of the helical reactor for many, many years, and I could take a guess, but it's been maybe 5 or 10 years, and I'm sure they've performed a lot of studies to show the optimal Dean vortices are being formed, and I believe that the helical reactor is very optimized, and I really don't see any further optimization occurring soon.
Okay, thank you. Makes sense. The next question here, is the lamp in the device helical, or is the fluid path helical?
This is Sherry again. So the fluid flows around a helical spiral wound flow path, and the lamp is rod-shaped, and it slides within the module, so the lamp is not helical. It's the fluid path which is helical. And as the fluid is flowing around the lamp, it is spiraling up from the bottom to the top, and it's spiraling and creating Dean vortices, which is creating the mixing, which is creating which is allowing the viruses to be equally treated by the UV light.
Interesting. Okay, thank you for that. Our next question here, given the odd profile of virus sensitivity, what has been the response of regulatory bodies in contrast to PPV being the gold standard? Your thoughts on that?
This is Todd. I can take a quick stab at it. We have gone forward, especially in Canada, to say, you know, we're considering using this technology, and they were very open to it. They liked it, and they basically asked for a complete data set on that. For right now, we're doing adventitious agent testing, but they would like that replaced with this. So we'll find out in this next year of exactly what they feel is appropriate.
Okay, that'll be interesting. Oh yeah, go ahead.
Let me add a little bit to that. Typically, no one step is effective for all viruses, and so your viral clearance study is assuring the safety or the absence of viruses by adding up a number of different steps. Some are better for one virus, some are better for another virus. There really isn't any step in existence that's equally effective on all viruses in the same condition. So the fact that this procedure is better for some viruses than others is really no different than any other step. One of the big advantages of UVC is that it does contribute an effective step for inactivation of parvoviruses. And while other viruses may be less sensitive, still, as Todd has found, there is valuable degree of inactivation of other viruses as well.
Okay, thank you for that, both of you. Another question here. At what scale were the virus inactivation studies performed?
I'll comment on ours. We did each study with approximately 150 mils of process intermediate solution.
And this is Kate. That's about the size that we're working at. Because of the size of the reactor coil and the pump flow rate, the minimum volume that we can actually work with is and process through the system is on the order of 100 milliliters. The first portion and the last portion we discard because they haven't necessarily been exposed to the full amount of radiation. So we typically discard the first portion, collect the middle portion, and then discard the last portion. So while we'll start with 100 mils, we'll end up with something more like 50 or 60 milliliters.
Right, okay, thank you for that. Our next question here, how involved is the maintenance for the unit?
This is Sherry Dolan. The systems are very simple and consist of basically a pump. The lab scale system consists of a Watson-Marlow peristaltic pump, and which has very low maintenance. The lamp is good for 1,000 hours, or we recommend you change the lamp once a year. There are a couple sensors, calibrated sensors, that need to be changed or calibrated once a year. So the maintenance is very simple. And the same goes for the process scale system. You just have a pump of your choice. You can use whatever type of pump you're comfortable with. And same thing, the lamps can be changed once a year, and the sensors need to be calibrated. So it's very simple operation. There's not a lot of maintenance, and there's not a lot of equipment that can break down or that you can have problems with.
Right, great to know. Okay, thank you for that. Our next question here, a little more involved. This audience member says, my protein solution product is a mixture of enzymes with various characteristics. Should we start by doing 200 to 2,000 joules per meter squared and check for visible damage, for example, aggregates, then activity, and settle on a dose that has the least effect and move to calculate residence time, etc.? Your thoughts on that?
This is Todd, and I'll say, you know, even at 2,000 joules per square meter, you're not going to see any visible impact to your stream. Like even if we looked at something like turbidity, we didn't see changes. So you have to go down and be thinking you're going to get only a few percent of aggregates if that's the mechanism that you're looking for there. So you may have to go to techniques that are a little more sensitive to see that. But I certainly would recommend, you know, picking a few doses and looking at for an enzyme if you have an activity assay, things like that may impact it.
Right, okay, thank you. Another question here. How is the UV intensity of the lamp measured, and how does the instrument adjust for the aging of the lamp?
This is Sherry Dolan. The intensity of the lamp is measured by the lamp sensor, and for the lab scale system, the measurement is in milliamps, and for the process scale system, the measurement is in watts per square meter. So those values are directly entered into your calculation spreadsheet, and your dose is based on the actual lamp reading.
All right, thank you for that. Our next question here. How can I determine whether my protein product is stable to UV inactivation? And a follow-up question, how do I determine what dose is appropriate for my process?
Do you want to start with that, Sherry?
Okay, this is Sherry Dolan. So as Kate had mentioned, Sartorius has systems that may be rented, so you can bring a lab scale system into your facility. And we normally recommend that you test at least 3 different doses, as Todd has tested, a low dose, a medium dose, and a high dose. and do all your analytical assays to determine your protein stability and determine whether or not your product has been affected. And from there, you would do exactly what Todd did. So I'm just going to pass this along to Todd.
So I think anybody that's making a product and they're in clinical trials, they have a pretty good analytical support package for understanding their product and characterizing it. So you got to have tools to take a look at that, and then knowing that the mechanisms of degradation are at the amino acid level and can be charge variants and things like that. If you can do some peptide mapping and see changes in there, those are the big things to see, where the damage is occurring, and then how to relate that to the quality attribution. I mean, really, your toolbox has to be good to be able to define and understand what's going on. Certainly, I would suggest, as everyone has said, do a range of doses and see where it is and understand. When we started this, we didn't really have a lot of information on what kind of dose was needed to kill the viruses. So I think you can pretty much zero in on what it's going to take based on the data you've seen today. You probably want to be up between 200 and 300 or 400 joules per square meter and work in that range and see if it impacts your product adversely.
All right, thank you for that. Our next question here. This audience member says, 2 suitable steps in my process for insertion have 5,000 liters upstream, low protein, or 500 liters downstream, high protein, equaling 150 milligrams per milliliter. Which steps is suitable in your view?
This is Kate. I'll start, and then the others can chime in if they want. It's probably either one could be suitable. Depends on what the low protein concentration one is, but the protein concentration itself in an upstream step may not be the major contributor to the absorbance. So it's probably a good idea to look at the exact details of each step and do the calculations in the calculator spreadsheet that Sherry mentioned and figure out what dosage ranges are available given the process steps you're talking about, and then to do some ranging experiments such as what Todd has just described in answer to the last question. And then based on the results and of the irradiation and the dosages and properties of each product stream, determine which one is the more favorable for use.
Yeah, I'd say check your A254 and ask Sherry to send you the calculation sheet, and you can plug it in and see what kind of flow rates you're going to need, and then you can calculate process time and throughput, and you'll know more about your hold times and if you have the capacity to hold the product in your manufacturing facility. So it's going to depend on your product, but it's not a hard study or just a calculation to figure all this out if you know the A254.
Right, that's exactly correct. That based on your A254, there's going to be a certain range that you can treat your product, and the best idea is to treat the lowest range, the highest range, and something in between. And you may be limited. You may want to reach a lower range that is not possible because your absorbance of 254 is too low, for instance. So that's exactly how you would go about doing that.
All right, thank you. Our next question. What model do you recommend for treatment of 20 liters?
I can take that. So we have the lab scale system, which I showed you pictures of. We have a GMP version and a non-GMP version. So the flow rate range is between 6 and 20 liters per hour, and depending on the dose and your absorbance at 254, if you were running 20 liters per hour, it would take you an hour to process your 20-liter batch size. So you may be able to treat up to 100 liters, 150 liters of product. It all depends on how long you're willing to wait for the processing to occur.
And yeah, I'll kick in. At our process full scale right now, where we have it inserted, we're looking at dealing with somewhere on the order of 30 to 40 liters of material, and we have the GMP small scale unit, and we'll be using that.
Okay, thank you for that. And on that note, another question here. Can you elaborate on the requirement to throw out the beginning fraction and later fraction in the UVC treatment, and how would this affect the step yield in a process?
Well, this is Kate again. The beginning and the end of the material is entering, the beginning of material is entering an empty tube. So the optical properties are not exactly identical, and this is the reason why we would throw out the beginning portion and correspondingly the end portion for the same reason, because after the material there's an empty tube again. In our smallest scale method that we're using for viral clearance evaluation or analytical observation, obviously you're throwing out nearly half of your material, but in a process where you're processing 20 liters, you're throwing out 20 or 30 milliliters. This is completely insignificant to the step yield. So it's a very different situation between doing analytical experiments and doing it in the context of an actual manufacturing process.
Okay, thank you. Our next question, do you recommend changing the lamp once a year with a low or high usage?
The lamps are useful up to 1,000 hours. That's a lot of hours to be used. So we recommend that you change the lamp either at 1,000 hours or after 1 year of usage. UV bulbs can last a really long time, but they're fairly inexpensive, and just to be on the safe side, we do recommend that you change it once a year or after 1,000 hours.
All right, thank you for that. Another question. Is anyone using UVC inactivation in a production process for a commercial product?
This is Todd. I know of a company in Canada that makes flu vaccine for distribution in the U.S. and other markets, and they have a UVC step in the process.
And this is Sherry. I have a comment that the first commercial scale UVC Uvivitec implementation was approved by the French regulatory agency in 2009, and this system is at Bayer Healthcare, and it produces a product called Hero Cream.
All right, great to know. Thank you for that. I think maybe one last question here. What is your opinion on using this treatment on FBS as a raw material?
This is Kate. That's certainly a potential usage. There's a couple of caveats that you want to consider. First of all, obviously, FBS is very high protein concentration, and the protein itself may contribute quite a bit of absorbance at 254 nanometers. So there may be limitations to the dosage you can achieve. Also, FBS is not particularly purified. If there's any circulating nucleic acids, that also will contribute to the absorbance and limit the dosage that can be achieved. And furthermore, as those of us who use FBS have seen, there's often a certain amount of sediment in the FBS, which makes it less than transparent, less than completely transparent. So that also contributes to the effectiveness of the FBS. I wouldn't rule it out. I think it's potentially worth exploring, but it also would be a good idea to check the FBS for its support of cell culture after irradiation, because with a large number of components in serum, you don't know whether you could potentially be inactivating some factor that's critical just because it happens to absorb UV radiation.
Interesting. Okay, well, thank you very much. We had a lot of great questions today. If you do have any further questions for any of our speakers, their emails are showing on your screen right now, and you can contact them via email. Thank you, everyone, for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event, and a survey window will be popping up on your screen, and your participation is appreciated as it will help us improve our further webinars. Now, please join us in thanking our speakers in the chat box, Dr. Kate Bergman, Dr. Todd Tallarico, and Sherry Dolan. We hope that you found this conference informative today. Have a great day, everyone.